Hello, my name is Saad Usmani. I am the Chief of the Myeloma Service at Memorial Sloan Kettering Cancer Center, and I'm here with blood cancer today. We are here in Chicago for the ASCO 2023 annual meeting, and we're going to be talking about all the exciting data coming out in multiple myeloma. We'll cover the newly diagnosed setting, some of the approaches to early as well as late relapses in the context of new novel immunotherapies, including bispecifics and CAR Ts. So I'm looking forward to having a wonderful, engaging conversation with my colleagues. I'll let them introduce themselves.
Hi, I'm Yi Lin. I'm from Mayo Clinic in Rochester, Minnesota.
Hi, I'm Shambavi Richard. I'm from Mount Sinai, New York.
Hello, I'm Meera Mohan. I'm from the Medical College of Wisconsin.
We now have real-world data with Ide-cel that was actually presented and subsequently published at ASH, and now, at ASCO, we have the Cilta-cel real-world experience. So, Dr. Lin, what are the, similarities and contrast with both those experiences?
No, absolutely. So it's very exciting that we're seeing here at ASCO for the first time real-world data from Cilta-cel. And of course, recognizing challenges all centers have had in terms of getting the patients on. So knowing these are somehow, very sub-selected group of patients. But we all know the patient that were on the waiting list, oftentimes they were on because they couldn't get on a clinical trial. They didn't qualify for a number of reasons. So it wasn't surprising to see in the Cilta-cel population, similar to Ide-cel, that a very high percent of those patients who received Cilta-cel in the real world also would not have met CARTITUDE-1 criteria. And so it was actually reassuring to see that the out-of-spec rate in the real world was 19%, which is right on par with the 18% on the label. So recognizing that the decision on the commercial specification for the CAR T is an FDA decision that sets a threshold. So even in CARTITUDE-1, even though we know from the trial conduct, manufacturing was successful, but if we apply the commercial specification, the out-of-spec rate was 18%. So in the real world, despite a very high percent of patients having demographic features that didn't meet criteria, the manufacturing, the characteristics, if you think about it, of the product were actually very similar to what was used in the CARTITUDE-1. Of course, we need longer follow-up to know, do those products really perform differently or similarly than the in-spec product? So there is an EAP study that has finished accrual, so we'll see the data in the future. Of the real-world data set, it was very encouraging to see, similar to the Ide-cel experience, despite these patients having various features of disease characteristics and demographic characteristic that didn't meet the study criteria, that the overall safety signal seems very comparable in terms of cytokine release syndrome, neurotoxicities. And we still do see that, unfortunately, but they were not excessively higher than what was seen before. The follow-up is still quite short, and so we know with CAR T therapy and myeloma, the response can take time to deepen. And so I don't think we've necessarily seen the deepest, the highest, best response rate yet, but the overall response rate was quite high and very encouraging. And so I think we'll continue to follow this patient population to see, in terms of durable progression-free survival, how did that look in the real world, which we know with CARTITUDE-1, that's what was really exciting, right, with the product. Wasn't just the high percent of deep response rate, but the very long PFS that we're seeing with a one-time dosing of the CAR T.
No, I love how you segued into that because, you're going to be, talking about that data, at this meeting as well. So tell us a little bit more about it.
So, it's very exciting. We've seen the primary analysis on CARTITUDE-1, and at that time it's a follow-up, median PFS not met. And then we've seen last year, longer follow-up with a median not quite 2.5 year, and a median PFS still not met. And so this year we finally have 3-year follow-up. So now we see that the median PFS is just a little bit short of 3 years, 35 months for all patients. but patients who achieved stringent CR higher than that, 38 months, and patients who had sustained MRD negativity, 6 months, 12 months, the median PFS has not been met yet, and median OS also not met. So very, very exciting data.
No, absolutely. I think we are seeing consistency with other, CAR-T2 cohorts as well. So again, the CART4 data, I'm really looking forward to, hearing, Binod share a lot of details with us. Okay, so what has your experience been in getting the product back? Whether for Ide-cel or Cilta-cel, is it the same compared to what was reported in clinical trials, or what has been your experience in the commercial product? Meera, we can start with you.
The clinical efficacy is, pretty comparable to what has been shown in the KARMA study with Ide-cel. With Cilta-cel too, so far we've the few patients that we have done the commercial CAR, I think the responses are pretty what is expected.
Okay. And then, with both the trials, it would take roughly a little over 4 weeks for us to get the product back after apheresis. Are you seeing the same thing, or there are delays?
I think at least at our institution, we do have delays with both Ide-cel and Cilta-cel. And obviously with Cilta-cel, we've had a lot of out-of-spec issues and delays.
Okay.
Yeah, that's been our experience as well. And as you know, that one of the challenge could be all that coordination, scheduling. At our center, I would say quite a high % of our patients don't live locally. We're in a very rural part of the country. And so we've just systematically said everybody plan to come back after 2 months instead of trying to time it on anticipated return. Now there's a delay and patients have to reschedule everything. We have to reschedule everything. If we do 2 months, it's kind of easy. So that's 2 cycles of whatever bridging therapy, and just plan on that. And if long as they're clinically stable, if the product come back a little bit early, that's great, but we're not going to reschedule everything. A little bit like transplant, how we plan for transplant. But if there's some delay, then hopefully that less people that we need to contact to reschedule, because yes, unfortunately, that still is an issue.
So, so consistency of getting the product back, vein to vein time is something that, we recognize as an issue.